Thank you very much, Ian, for your kind introduction. I remember 14 years ago when I first met you and Austin. Austin and Ian came to my lab, and I talked about knockout mice of now called Nanook. Austin didn't change his face at all. Poker face. It's interesting. But Ian got pale. Silent Ian is a very honest person. Thank you. So anyway, 20 years ago, 1996, I finished my postdoc training in San Francisco, and I came back to Japan. And I was struggling to deciding what I should do as an independent scientist. And I was losing my confidence to continue my science. Within 6 months after I came back to Japan, I became sick. I suffered from PAD. There must be many physicians in this room, but I'm sure you don't know what PAD is, because we named this disease by ourselves. PAD stands for post-American depression. So I really suffered from this depression, and I was about to quit science, and I was about to go back to clinics because I was an orthopedic surgeon. But soon after, in early 1997, I was astonished by a paper in Nature. I was so excited. That was Dolly. When I found this paper, it was covered by all the Japanese TV press everywhere. Even my mother knew about Dolly. So, I was so excited that I was able to overcome my PAD. And by being inspired by Dolly and by being encouraged by Dolly, I started a new project, which resulted in iPS cells 10 years later. So in 2006, from mouse, adult, and embryonic fibroblasts, we became able to induce ES-like stem cells, which we designated iPS cells, induced pluripotent stem cells. The procedure was extremely simple. All we need is a combination of 4 factors, transcription factors. Next year, 2007, we and multiple other groups was able to recapitulate same reprogramming in human iPS cells. Initially, we used skin fibroblasts, so we needed to perform skin biopsy. But now, we can make iPS cells from many types of somatic cells, including peripheral blood cells. All we need is a very small amount of blood cells, just 5 ml or 10 ml. By introducing those 4 factors, actually now we have been using 6 factors, we can convert blood cells into iPS cells. Once they become iPS cells, we can expand iPS cells as much as we want. We can have hundreds of Petri dishes containing iPS cells from a very small amount of blood cells. After expanding iPS cells, by treating these cells with some cytokines and growth factors, we can convert iPS cells into many types of somatic cells. For example, we can make this kind of beating cardiac muscle cells, hundreds of Petri dishes containing beating cardiac myocytes. So now that we have this technology, we are trying to bring this to patients. There are 2 major medical applications of iPS cells. The first one is cell therapy. For example, if there is a patient suffering from heart failure, he or she cannot wake up, he or she cannot run, all we need is a small amount of blood cells from him or from her. We can convert blood cells into iPS cells. We can expand iPS cells as much as we want. Then we can generate cardiac muscle cells from iPS cells. The patient is very sick. His or her heart cells inside his or her body is very sick. However, these new cardiac myocytes from iPS cells are not sick. So by transplanting those newly generated cardiac muscle cells back into the patient, we should be able to recover the function of heart in patient. That is cell therapy. But we have another very important application of this technology. We will not transplant cells back into patients, but we will continue using those cells in lab, in Petri dish. By using patient-derived iPS cells, we can make disease models, and we can perform drug screening. We can use them in drug discovery. So both of them are equally important. Six years ago, 2010, Kyoto University decided to start a new research center exclusively for iPS cells, Center for iPS Cell Research and Application, CIRA. I have been directing this institute for the last 6 years. We have a very clear goal or vision or mission. It is to contribute to new therapies using iPS cell technology. So let me introduce you a few examples of each of the two major medical applications. Let me start with cell therapy. There are many applications which are getting closer to clinical trial, such as Parkinson's disease, macular degeneration, spinal cord injury, cardiac failure, platelet deficiency, and so on. We are hoping that within 2 or 3 years, we can initiate clinical trial. Among this, macular degeneration is the most advanced. Actually, Dr. Masao Takahashi, she's now in Kobe. She started clinical trial already for patients suffering from age-related macular degeneration. So she generated iPS cells from patients on skin cells, and from iPS cells, she generated retinal pigmental epithelial cells. She made a sheet of pigmental epithelial cells, which is shown here. And her team replaced patient on injured retinal cells with newly generated retinal epithelial cells from iPS cells. It's been 2 years, and I heard from Dr. Masao Takahashi that patient has been doing very good without any major side effect, including tumorigenicity. We don't see any tumorigenicity. But so far, we have been able to perform operation only 1 patient. Why is that? Because it was autologous transplantation. Autologous transplantation is a huge advantage of iPS cell technology. But in reality, making iPS cells and also expanding iPS cells, differentiating iPS cells into retinal pigmental epithelial cells, and performing rigorous quality control checks is actually very, very expensive. And also it takes at least several months. Actually, it took almost a year in the first case. And it costed us almost $1 million just for one patient. Because we did a lot of whole genome sequencing and many other testing. That was the major reason why we did only one patient. Instead of autologous transplantation, we are now trying to shift from autologous to allograft by establishing iPS cell stock for therapy. In this strategy, we are making iPS cells in advance, not from patients, but from healthy volunteers or from cord blood samples. Because we make iPS cells in advance, we have enough time to perform a rigorous quality control to select the best and safest iPS cell clone from each donor. But this is not autologous. We have to worry about immune rejection. In order to minimize immune rejection, we need to match HLA. But as you know, HLA is so diverse. None of 200 people in this audience have the same HLA unless we have identical twins in this room. But we are making iPS cells from super donors. Who are super donors? By collaborating with Japan Red Cross and also Japanese cord blood banks, we can now efficiently identify these super donors. So once again, who are super donors? Super donors are HLA homozygous donors. So in this slide, I show the HLA haplotypes of 10 patients, 10 recipients with different colors. We receive one HLA haplotype, one color from our father, and another different one from our mother. It's a combination of two, which make HLA haplotypes more diverse. So none of these 10 recipients, patients, have the same color combination. If we want to prepare iPS cell stocks that can match these 10 recipients, we need to generate iPS cells from all of these 10 individuals. It's a lot of work. However, if we identify just one HLA homozygous donor, and if we make just one high-quality iPS cell line from just one HLA homozygous donor, we can perform cell therapy into 4 out of 10 recipients. Because these 4 patients have red and something else. As long as he or she receives red from HLA homozygous donor, he or she cannot distinguish transplanted cells from his or her own cells. According to this model, we have calculated how much HLA homozygous donors are required to cover most of our population, Japanese population. And we found it's not thousands. All we need is approximately 100. By having 140 HLA homozygous donors with different colors, we can cover up to 90% of all the Japanese population. By having 70, we can cover 80% of all the Japanese population. And if you look at this point very closely, by having just one HLA homozygous donor, with the most frequent HLA haplotype among the Japanese population, just one HLA homozygous donor, we can cover up to 17% of all the Japanese population. So this is our primary goal. For clinical trial, this may be sufficient. And actually, by collaborating with Japan Red Cross and cord blood banks, we have already identified many HLA homozygous donors. And we have generated GMP-grade iPS cell lines already from the first HLA homozygous donor who has the most frequent HLA haplotype in our population. So we generated 40 clones, and we performed various tests of these 14 clones, and we selected 2 best clones. And we have shipped these 2 clones last year, August 6, 2015. When we first shipped our first GMP-grade iPS cells, we have a party. We took a picture. The cell is here. It's not as fancy as Dolly, but we're so happy. But the point is, there are more than 50 people. So we are learning, making GMP-grade iPS cells is a lot of work. Making research-grade iPS cells is still a lot of work, but one hardworking graduate student can make it. But making GMP-grade iPS cells is totally different. We need 50 or more people. So this is our plan. It's a 10-year project. In the first 5 years, we would like to make like 10 to 15 iPS cell lines so that we can cover up to 50% of all the Japanese population. In the next 5 years, we want to generate as many iPS cells as possible so that we can cover up to 80 to 90% of all the Japanese population. Oops. So let me briefly talk about this second application, drug discovery. So let me use this intractable disease as an example, or achondroplasia, a form of dwarfism. So this patient is suffering from dwarfism because he cannot make chondrocytes properly. He has a point mutation in FGF receptor. We don't have cure for these patients. Dr. Noriyuki Tsumaki, one of my colleagues, he generated iPS cells from the patient and also from normal individual. When undifferentiated, he did not see any differences between patient and normal individual. However, when he induced differentiation into cartridge, he observed marked difference. From normal or individual iPS cells, he was able to generate a beautiful cluster of chondrocyte, which is stained in red. However, by applying exactly the same procedure, he failed to generate chondrocyte from patient iPS cells. So this means we can recapitulate the disease in Petri dish. Patient cannot make chondrocyte. Patient iPS cells cannot make chondrocytes. We have only one patient, but we can make hundreds of Petri dishes containing patient iPS cells. And in each of hundreds of Petri dishes, we can test different drug candidates. It takes only, I'm sorry, I'm going to finish very quickly. So by using this kind of patient-derived iPS cells, we can promote drug discovery tremendously. And he tested many drugs, especially existing drugs. And to our surprise, he found that statin, which is a very famous, popular cholesterol-lowering drug, can make a huge difference. By just adding statin into patient-derived iPS cells, now he can make a beautiful cluster of chondrocyte cartilage. Because statin has been in clinic for the last 20 or more years, it's been widely used. I believe probably 20 or 30% of you are taking statin, probably. We can quickly bring this research result into patients. So that's what we are now trying. For many other intractable diseases and rare diseases, this is the strategy. Making disease models by using patient iPS cells and testing thousands of existing drugs to promote drug discovery for rare patients and other intractable diseases. So once again, we are doing our best to realize these two major medical applications. We have more than 500 people in Kyoto, or more than 30 groups. And hopefully, in the next 10 years, 20 years, we can help, we can contribute to new therapies by using iPS cells. So thank you so much for your kind attention.
Thanks very much, Shinya. And I need also to thank the company of biologists who sponsored Shinya's presentation today. So do we have some questions? Andrea?
Thank you for your presentation. I have a question concerning your HLA data. Japan is an island nation that has been isolated from the rest of the world for many centuries. Does this simplify the diversity of HLA variants within the Japanese population? And if you think about less isolated countries or populations, or like United States with immigrants from everywhere, how does this affect the frequency of the HLA that you call super?
That's a very important point. Probably US is one of the most diverse countries in terms of ethnics. So they have many ethnics, but they have like Asian ethnic, Caucasian ethnic, African ethnic, and Spanish ethnics. And we need to prepare HLA stocks for each ethnic group. So in Japan, we only need 150. But in the States, probably we need like 500 or 600. But it's not like thousands. And in UK, the number should be very similar to that in Japan.
I was involved for some years with a project on alternatives to animal testing using human embryonic stem cells. And in those days, iPS cells had only barely begun. And do you see this as another possible use in drug discovery as alternatives for using animals?
Yes.
What would you say are the prospects within this field?
So, for example, many animals are being used to test cardiac toxicity of newly developed drugs. But I think we can replace many of them with iPS cell-derived human beating cardiac myocytes. So I really want to reduce the number of animals. Actually, Kazu Takahashi, who was the first author of the two iPS cell papers in Cell, he one day said to me that to him, the biggest motivation is to reduce animals. To me, the biggest motivation to use them in hospitals, but he has his own motivation. I didn't know that.
person on the Animal Welfare Committee for Roslin, so I would say both are important.
Oh, yes, yes.
We have a question up here.
In terms of, is this on? In terms of using iPS cells as models for disease, when your disease involves complex interactions between different cell types, and I'm thinking particularly of neurodegenerative diseases here, how useful do you think they will be as models of disease?
Yeah, so the example I showed is very simple. Cartridge, just one type of cells. But as you just mentioned, many diseases are more complex. So we need to prepare more complex disease models. But nowadays, many scientists have been trying to generate like mini organs, like mini brains, mini intestine. So by applying those kind of 3D approaches, we can mimic those more complex diseases, at least to some extent. But they are just cells still. You know, iPS cells are not Dolly. So we need to use both animal models and cell models like iPS cell technology. We need to combine the two.
Kirsty, there's a question in the middle here.
Hi, thank you for a great presentation. So the work done creating iPS cells from healthy members of the public is obviously, well, presumably really important, because cells that are derived from healthy members of the public are probably going to be less prone to then degenerating again, as you know, if you took them from somebody who already had a faulty genotype in some way. And I was wondering whether what screens are done when selecting members of the public to take cells from, because the HLA type is presumably not the only factor which is important when regarding the health of a cell that then develops in a diseased patient. And I wondered whether, whether the patients are screened for genotypes related to disease, or for genotypes which, when put in the environment of a patient who has, you know, perhaps a condition caused by complex genotype, there's lots of other things going on there, and it's just not a very healthy environment, that perhaps cells from a healthy person might then react badly and become diseased in that environment. I wondered what other selection processes there are.
Yeah, I can't agree with you more. It's very important. So in the case of HLA homozygous donors, they are either from platelet donors or cord blood samples. So in the case of platelet donors, they are already adult, and we know they are healthy. So, and many of them can repeatedly come back to our hospital so that we can make it sure that they will remain healthy. But in the case of cord blood, it's very difficult to trace their healthy condition. But cord blood has one big advantage. It's from baby. So in terms of a mutation acquired after birth, the cord blood sample is much, much better. But we cannot take record of donor, him or herself. So it's kind of balance between the two. And for research purpose, like for drug discovery purpose, it's also important to have iPS cells from healthy population. So we have been collaborating with some major Japanese company. They have their own healthcare system of their employees and family members. So in Japan, we still have a very long employment system, 50 years or even longer. So they have a very good health record of many of their employees. So by collaborating with those companies, we can now making iPS cells from healthy volunteers.
Thank you.
A question over here.
The statin result over here, the statin result is very striking. Do you know if the mechanism through which it's working is through interfering with steroid hormone signaling?
Yes, so we are trying to understand. Dr. Tsumaki has been trying to understand. And by using iPS cell model, he now has some clue why statin can actually have a positive effect. Well, it's still unpublished, so probably I shouldn't tell. But he is having a very good clue.
Sure. So on that subject then, statins are normally given to older members of the population where steroid signaling is perhaps less important. But the implication here would be you would give it to very young developing children.
Exactly.
So
yes.
what are the implications for statin administration in young children interfering
Oh,
with
that's
with
what
sex
we
driven?
have been being asked by our regulatory person. But many pharmaceutical companies have data about safety tests of statin even in children, like 5 years old or 6 years old. But we want to apply this to like 2-year or 3-year-old patient. So that's what we have been struggling with. You know, PMDA is FDA or EMA equivalent of in Japan, and they asked us to provide data with 2-year-old, 3-year-old baby, but we don't have any data. And if we try to collect those data, it will take many years. So maybe we should start with like 4-year-old children instead of 2-year-old. Yes, it's very important.
Okay. Before we move on, I'm going to ask the last question. Actually, it's 2 questions, and you can respond very briefly to them and maybe longer later. But we were all very surprised when you announced the 4 magic factors and Nanog was not one of them. So my first question is, were you surprised? And my second question is, what did you say to Kazu when he told you that this was the 4 and Nanog was not one?
So we started with 24 candidates, and of course, Nanog is one of 24. So I was very disappointed when we found Nanog is not required. When I first learned from Kazu that we are getting ES-like colonies from fibroblasts, I told him it must be some kind of mistakes. So you should repeat the same experiment. And he did many times, and all the time it worked. So I gradually become believed our own result.
Okay, right. Let's thank Shinya again for a great talk.
Thank you.